Thank you. My name is Allie Welsh, and I'm really honored to introduce our first speaker, Dr. Mike Angelo from Stanford. He's going to speak to us about new technologies for molecular imaging studies, specifically multiplexed ion beam imaging. So we're honored to have him, and join me in welcoming him. Thank you.
Thanks for having me. My lab has been working at developing a multiplexed imaging technology that utilizes antibodies for a multiplexed IHC assay where each of the antibodies is labeled with a mass tag instead of a fluorophore or a chromogen. That technique is called multiplex ion beam imaging. And so, my lab kind of focuses on sort of broadly the development of new bioanalytical platforms, and at the moment, that's focused on multiplex imaging assays, and we like to use those assays to look at, to explore sort of normative distributions of immune cell populations in solid tissue, and also to look at how those immune cell populations are perturbed or deviate from these sort of normal distributions in different disease states. So today I'm going to kind of quickly go through the working principles of the instrumentation platform, and as much as I would like to, for the sake of time, I will not dwell too much on the technicalities of how the instrument works. And I would like to just kind of get to the end. Unfortunately, where I don't have a ton directly related, or related at all for that matter, I guess, to neuroblastoma, but the the reagents that we're developing is particularly for looking at different immune cell subsets in solid tissue. We're starting to deploy those now, looking at different pediatric solid tissue malignancies, including neuroblastoma. And I hope, within about a year's time to done similar analyses in that arena as well. So, so to start off, so MIBI, like I was saying, it's essentially a high-dimensional imaging platform, and the sample prep, I like to go through talking about just how the samples are processed and what happens when they go in the instrument. And so everything we do is with formalin-fixed paraffin-embedded tissue, and we mount it on a microscope slide. And so the reason why we try to build our assays and our reagents around FFPE is it makes sample procurement much easier than if you have to use fresh frozen. You can use archival clinical cohorts, which is everything I'm about to show you is from archival human tissue blocks. So we start off with the tissue on a slide, and then the tissue is treated with a single antibody master mix. It has all the primary antibodies, and each one of the antibodies is tagged with a unique elemental reporter. If you're familiar with CyTOF, the elemental labeling of the antibodies is analogous to that. So it's not a cyclical process, so the entire panel, 30, 40 antibodies is stained at the same time. It's then put into the mass spectrometer that we use, where an ion beam comprised of charged oxygen particles, or xenon particles, or gold particles, there's a bunch of different beams you can use, is focused down to a very small spot, and that beam is moved pixel by pixel across the tissue, and as that happens, secondary elements are liberated, and those are extracted into a mass spec. This is just kind of showing a cutaway of our instrument. And so essentially it goes around this 90-degree bend, which selectively enriches for the elemental reporters that we care about over organic material that arises from the tissue, so that what goes into the mass spectrometer ends up being predominantly comprised just of elemental ions. And so when we look up in the region where in the mass range where the elemental ions occur in the mass spectrum, we essentially end up with a multiplexed image where for every pixel in this image, we have a mass spectrum associated with it, so that we can go through and look to see what the level of the original antibody in that specific pixel in the image was. So, for example, what you see here is equivalent to what we use instead of DAPI in fluorescence assays, and so this is an antibody for double-stranded DNA, and it's labeled with a metal, a specific isotope and metal called yttrium-89. So, for instance, we were reading this out, we would go and we would look at the peak that was at mass 89, and we would make an image only comprised of the ions that occurred within that peak. So, this is one of our favorite specimens, which is tonsil. lots of tonsillectomies out there, which is good, keeps us in business. So it's usually not hard to get tonsil, which is a nice thing because we need it for a lot of our QC. So probably the most important thing with MIBI, because it's an antibody-based platform, I like to bring out the fact that, it's by no means a magic box. And so, if you start off with crappy antibodies and crappy staining, you're going to get crappy staining out with MIBI. So, probably one of the things that we spend the most amount of time doing is QCing the reagents and making sure they work very well, usually first in single-plex assays. So every primary antibody that comes into my lab, I make everybody, even though they kick and scream about it, they have to do, chromogenic stain without derivatizing the antibody first to see if it works. If it does work, then we metal conjugate it, and then we test it again under the same titers to make sure it still works. And then if it does, we move it on to the multiplex panel with a MIBI analysis. And 95% of the time, if we have a primary antibody that's metal conjugated that works well with chromogenic IHC, it works great when we get it to this stage. So that's kind of abbreviated how we look at the reagent process. But when we are QCing these, we look for appropriate co-expression of markers, and that they're histologically located in the right place, and that two things that we know should not be co-expressed do not occur in the same cell. Just kind of run a few examples of that. For instance, if you're looking at tonsil, B cells and T cells should sort of in a mutually exclusive way, express CD3 and CD20, or so 20 on B cells and 3 on T cells. So we see that here, just kind of flipping through these for the sake of time. I would love if anybody just wants to look at pictures, catch me after. We look at pictures all day. But just flipping through these, essentially with each one of these markers, we focus on making sure that the immune cell phenotypes that we're seeing are within the realm of what we know to be correct, both from a histological location and on a single cell level on a sort of multiplex phenotype. So then the sort of general performance metrics is, we can adjust the resolution across a pretty wide range, about 100-fold range. So we can go down to 100 nanometers or up to 10 microns. And probably the first question that I would ask in that situation is like, well, why not always do it at 100 nanometers? And the answer is because it would take forever. So I'll show you sort of one benefit of being able to go up and down on the resolution range in a second. But the field of view size, also we can adjust pretty dynamically from small as a micron if we wanted to, up to like 1.5 millimeters. We can do we have about 55 elemental channels in principle we could label and measure proteins with, although in practice we rarely do that many. Our sensitivity is around 1 to 5 antibodies per pixel. We can require around 10,000 pixels per second if we're going full tilt. So then, what's kind of the use of having the big resolution range? Well, I'm going to show you sort of two quick sort of use cases that we have in the lab. One is where we're looking at full tissue sections and sort of identifying region of interest. Excuse me. The other, which is the ones we try to do for the larger cohorts, is tissue microarray analysis. But in the case of large tissue sections, one nice thing with this is that with MIBI, you can actually you get more than one pass at imaging the tissue. So for each pass of the ion beam over the tissue, it takes off, since we're using standard 4 micron thick sections, it takes off maybe about the top 100, 200 nanometers or something like that. And that's usually enough volume that we can get pretty good signal to noise on most markers. So what that means is that we can go and we can scan a full tissue section at lower resolution and go very fast and acquire all of our antibody markers. We can go and on that low resolution survey scan, we can go and identify regions that we want to do follow-up medium or high resolution scans, and then we can rescan those subsequently afterwards. So to give you an example, here's like a tissue section that's about 1 centimeter by 1.5 centimeters or so. And you can see this is an overlay with keratin, vimentin, and smooth muscle actin. And so these two regions, from first glance, they look pretty similar, even on the 3-color phenotype. They're keratin positive, more or less SMA and vimentin negative. But as we start to look through the panel, we can see they actually have some unique features about them. So, for instance, the region on the right has, that's your right, yes, the right is beta-catenin high, whereas the one on the left is beta-catenin low, and that is accompanied by sort of what looks like a loss of HLA class 1 expression on the right. And now we go and we look to see sort of where different immune cell subsets are. The one that has retained class 1 expression has a significantly higher number of CD4 and CD8 T cells within the near vicinity, as well as dendritic cells and macrophages. And so then we went and after this, we identified some interesting regions that were beta-catenin high and went back and did medium resolution scans on this same tissue section. And so you can see in green the immune cells that's studying with CD45, and then the red the tumor that's marked by beta-catenin. And so the great thing about this, even at a very low level, having multiplex analysis makes some of these distinctions that have been, I think, kind of difficult in tumor immune oncology. In terms of, like, for instance, with PD-L1, it's a little tricky because it can be expressed by tumor, but it can also be expressed by dendritic cells and macrophages, which tissue macrophages in particularly, depending on how reactive they are, they can have very funny-looking morphologies, and it can be very difficult purely based on morphology to be able to distinguish them from tumors. So one nice thing, sort of very straight off that you get when you look at multiplex data is that the kind of distinctions that you want to say, so for instance, is PD-L1 on tumor or immune cells, becomes a lot easier here. So for instance, on first inspection, it's pretty easy to tell that almost all the PD-L1 expression is on CD45 positive cells, and that those CD45 positive cells express CD11C, also not pictured here. They also express CD68 and CD16. And then there's some variable degree of IDO expression. Oh, whoops. Anyway, where is it? There you go. There it is. IDO expression in green. So, and now if we go up and we want to look at some of these markers at higher resolution, I'm just kind of showing this just to show what it looks like. In actuality, we hardly ever scan at images at this high resolution because the downstream analyses don't seem to really benefit from the higher res, so we usually stick at the medium resolution scans. But for the sake of demonstrating, it's kind of what they look like if you go to the higher res fields. So, 13 minutes, okay. So, the study that we did to show you with the tissue microarray data, one that we have recently submitted, hopefully will be out in the wild pretty soon, is looking at a cohort of triple-negative breast cancer tumors. This was patients that came in over a 10-year period at Stanford, and we analyzed we wanted to look at the immune infiltrates in these tumors using the panel that you see here, which is focused predominantly on immune cells. We have some tumor and stromal markers in there as well. And so just to give you an idea of what the data looks like from one of these. So each one of these was a millimeter tissue microarray core, and so we looked at around 42 of those cores. And in this case, we did like the 800 micron field, which is sort of the inscribed square of a millimeter core. And so here you can see sort of in one case where we have tumor region marked by beta-catenin, and then CD45 marking immune cells here. And then here's some different insets to show how we can see the co-localization of different immune cell markers and be able to identify sort of multiplex phenotypes. So the things we want to think about when we're starting to post-imaging analysis, right? And I think this is going to be the big area over the next few years that is going to have to be sorted out, which is what are the best practices for analyzing the multidimensional imaging data, and how do we draw inferences and see relationships that are true, that we can confidently say are statistically significant and are not simply an artifact of the prevalence of certain cell populations, so on and so forth. So, the first thing we want to do is we want to be able to define which cell populations that we feel like are functionally important that we can say are there in significant numbers. How are they spatially related to one another? And then lastly, how do these relate to sort of clinical endpoints? So the first step of that, we start with this image, which is just simply a 2D array of numbers as far as the computer is concerned. So we need to have more or less circles drawn around all of these different pixels to delineate what's a cell and what isn't a cell. And so that ultimately we can go from something like this to something like this, where we can, in a human interpretable way, be able to highlight with some kind of color coding scheme the different cell phenotypes that are present in the image. And so here's just an example where after segmentation, we've then taken all the single cell data for all the single cells that were identified and segmented out in the image. and then clustered them to identify different immune cell subsets, and then went back to that image and colored it to demonstrate, for instance, where B cells are. And so you can see here that there's this tertiary lymphoid structure with a big cluster of B cells. So the first step of that is drawing the circles. And so ideally, we don't want to do that manually, ideally. Unfortunately for Samir, who is a very talented high school student who decided he wanted a project last summer, he came, and he's going to be on the paper, so it worked out well for him. But he came in, and we've gone to something that we're very excited about, which is that we're using a machine learning neural network to be able to do the segmentation in a much more accurate way. To be able to use that, we needed training data, which is just simply a bunch of examples of manually segmented cells. So for that, Samir went and he highlighted, he went and outlined manually a bunch of cells kind of like this, which we then fed into this deep learning neural network. And essentially what that does is it comes out with a classifier to say, for a given pixel, is it an edge pixel on the nucleus, is it a central pixel, or is it a foreground pixel? And what we found was this actually, and most of this the platform that we utilized was something called DeepCell, which was developed by David Van Valen. He's new faculty at Caltech, and to his credit, we really didn't really do very much to alter from this platform he had already come up with. We put in the data, and the results that we got out was actually much better than anything we were able to achieve with all the other typical segmentation platforms out there. And what it really excels at, and I think this is really to David's credit for how innovative this is that it does much better at being able to get both really big cells and small cells at the same time and not make these mistakes where a lot of times with traditional algorithms, if you try to get the small ones, it wants to break the big ones up into like 4 or 5 pieces, kind of like you see here. And if you optimize it for the big ones, it'll just miss the small ones altogether, so on and so forth. So we're really excited about this. It's by no means perfect at this point, but based on how well it worked and how quickly it worked, we think this could be going forward, sort of a universal platform and approach to being able to get really accurate tissue segmentation, which has been elusive in the past. So once you have all the cells segmented, you then need to be able to group them in a computational way into sort of, what we feel like are functionally meaningful subsets. So we did that with, once you have the cell segmented, you can actually just integrate the total expression of each of the proteins on a single cell basis and get that into a tabulated table that has like the XY coordinates for each cell and what they're expressing. And the good thing is that once the data is in that form, you can feed it into some kind of, a lot of the existing very, well-vetted high-performing clustering algorithms. In this case, we used FlowSum, and so you can see that it clustered it. We were able to delineate all the sort of major lineage subsets of immune cells. And then once we have those, we can now map those cells back onto the tissue. And so just to kind of give you an idea of the accuracy of it, we're pretty pleased. So for instance, this one little orange cell here is supposed to be an NK cell, and you can see it's the only one that's CD56 positive. This one little yellow cell here, or 2, I guess, is supposed to be a B cell. You can see this CD20 positive, and then the Tregs that are marked, I think in, violet or something. They're, you can see they're FOXP3 CD4 positive. So, by and large, the accuracy of this approach seems to be pretty good. And so if we look at a high level then of all the tumors in the cohort, this is what it looks like. So for all 42 tumors, you can quickly see that there's a big variation in the number of immune cells, but also of the relative abundance of different immune cell populations within a given tumor between patients, right? So one of the very interesting things we saw immediately, though, was that there seemed to be a strong interdependence between the presence of certain immune cell lineages and the presence of others. So for instance, if you had any immune cells at all, if there were immune cells there were always macrophages, which isn't, I guess, probably too big of a surprise because, tissue macrophages or even in uninflamed states or, are resident in the tissue and they're usually there. But what we found then was that essentially we could tell you, for instance, if you told me that a tumor had NK cells in it, I could tell you with 100% confidence that that tumor would also have significant numbers of B cells, CD4s, and CD8s. Every single tumor had NK cells and it had all these 3 other lineages. And so you can kind of look at this and see that there's a stepwise recruitment that we saw that we didn't see with any of the tumors that had deviated from these trends. And in some of the tumors that we've imaged more recently of different types from triple negative, including like lung carcinoma and all that, we've actually seen the same trend where we haven't really seen much of anything where they've deviated from this. So then the question becomes, which cells are co-localized more often than you expect by random chance? And so this is, if I were to say, this is the thing, if there's anything I would like to say for people in the room to watch, because I think this is probably going to be the area of the most hazard for interpreting multidimensional imaging data going forward, which is to be properly conservative in the way that we phrase statistical questions with the data. And so what do I mean by that? So the abundance of cell populations can vary dramatically depending on the tissue. There may be no immune cells. There may be thousands of immune cells, right? There may be very few tumor cells. And so, the cell localization then can also be highly affected by the tissue architecture. So it's important that whenever you're asking, whatever question you're asking, is that it's taking into all of these predisposing effects of where cells might or might not be just by random chance because of how many of them are there or which tissue compartments they're just naturally restricted to. So the way that we did that is that, so to give you an example of trying to look at sort of the tissue-wide organization of tumor versus immune cells, the way that we do that is instead of, we want to do a pairwise distance matrix, but we don't just want to do it on the tissue as it exists. We want to compare it to something to have a base reference for what it means. So we essentially do around 1,000 randomizations where all the immune cells and tumor cells are randomized to different locations from where they were before. And we do that about 1,000 times to get a null distribution so that we can then calculate a z-score. And so that we then have a pretty good idea that if we see co-localization of certain markers for immune cells or for tumor cells, it's something that we can confidently say shouldn't have happened by random chance. So just to give you an example of that, what we found is on some patients, we see tight co-localization of immune and tumor markers and anti-co-localization of a tumor marker with an immune marker. And whereas in others, we see that there seems to be not really any real relationship between where tumor and immune markers are in the tissue. And so what we found was this seemed to correlate with sort of two main subtypes of tissue architecture, one being the compartmentalized phenotype, where the tumor cells are sort of all together and they're surrounded by seeds, immune cells, and the other is this mixed phenotype. And we were able to derive this simply from the distance score of the different markers. And this does, to some extent, relate, as you might imagine, to immune infiltrate, but it also has a strong degree of variance that can't be attributed to that. So why does this matter? Well, one of the main things that we found is very interesting was that the disposition and presence of different checkpoint molecules seemed to be directly related to whether or not these tumors were mixed or whether they were compartmentalized. So for example, again with PD-L1, which can go between tumor and immune cells, we found that if we're talking about compartmentalized tumors, PD-L1 was heavily predominated on immune cells. Whereas if we looked at mixed tumors, PD-L1 was heavily enriched in tumor cells. As a matter of fact, if we calculate then all of the number of PD-L1 positive tumor cells and compare it between mixed and compartmentalized, you can see there's a very big difference in tumor PD-L1 positivity between mixed and compartmentalized. Similarly, we also saw that IDO positivity in immune cells is strongly biased towards being in compartmentalized histologies rather than mixed histologies. And the other one that we thought that was very interesting was that CD4, the ratio of the number of PD-1 positive CD4 to CD8 T cells was also related to whether or not tumors were mixed or they're compartmentalized. So compartmentalized tumors seem to have an overabundance of CD4 PD-1 positive cells, whereas the mixed ones had an overabundance of CD8 PD-1 positive. So take that together. That means that the compartmentalized have or more often than not have a lot more immune cells that are PD-L1 positive and IDO positive. And if there's PD-1 present, it seems to be on CD4s. And on the mixed, it seems the PD-L1 seems to be on tumor, PD-1 seems to be on CD8s. So I'll kind of skip through this. I'm running out of time, but more or less we were just showing that if we can do this, we can also ask where these markers are more enriched at the tumor immune interface or in the bulk, and we're able to see some interesting trends with the acetylation versus methylation, where for instance the acetylation is much brighter at the border and diminishes as you go in. And we kind of looked at this to do a clustering analysis to see which markers were enriched at the border. And we found that in a portion of the compartmentalized tumors, the PD-L1 and IDO were highly enriched at the tumor immune interface, and that this was accompanied by aberrant expression of HLA-DR by the tumor, which diminished as you go into the tumor bulk. So the tumor itself was expressing class 2. And then lastly, what we thought was the most striking, which we weren't really expecting to find, was that there seems to be a pretty big difference in survival between patients with triple negative that have either compartmentalized or mixed phenotype. And because of time, I won't go into this completely, but we're thinking that some of the phenotypic features that we've seen in this may be indicative that the compartmentalized phenotype, there's something about the cells that are there that dictate they might be a chemo-responsive subset of triple negative. So with that, I'd like to give a special thanks to Liat, who did all of the wonderful computational analysis on this. Erin, who has her own work that she's doing on tuberculosis, but also contributed heavily to the reagent development here. And Mark, who's the instrument guru, and of course our funding agencies and other collaborators. And if there's any time, I can take any questions.
And we will have some time, hopefully, at the end of the session for more discussion. So one question, did I see a question?
Yeah, if you sample 3 different regions in a tumor, do you get the same cellular phenotype, or does it vary from region to region? And if it varies, then how do you conclude what the overall phenotype is for the tumor?
Great question. Yeah. I don't know. So that.
This could allow you to do that.
Yeah. Well, it could. I think that the real answer. I think the big answer to that is because probably one of the other questions that we often get is you need to do the whole section because then you'll see everything. And the thing I like to remind people of is that. We're always going to be woefully undersampling with imaging, always, no matter how fast anything gets, right? Because when you take out a tumor, right, somebody looks, and tumor's like, it's like a macroscopic object, right? And then the pathologist goes and cuts off maybe, 10, 20 tissue blocks, right? And then we take one of those blocks that's 2 millimeters thick, and we cut off a 4-micron section, right? So even if you're doing the whole tissue section, that's still like hardly anything of the total bulk. And so my suggestion on this is when this comes up is like, so then how do we fit this in terms of what we should be sampling? Your best bet is you collaborate with a pathologist that knows the tissue histology very well. You get them to screen like the whole case on H&E, right? And you get them to pick, however many fields they feel like best, captures, the variability that they're seeing. So, if it's 5 fields, great. If it's 10, then do that. And I think that's probably the best hope we have of trying to get at this. But it's tough. And to be honest, I don't have a great answer for it at this point.